The overall goal of this procedure is to develop a simple and efficient strategy for immobilizing any protein of interest on polyacrylamide hydrogels in order to independently control various parameters of the cell microenvironment. This is accomplished by first spreading the protein solution across the PDMS stamp surface. The second step is to carefully dry the structured surface of the PDMS stamp with a steady flow of high-purity nitrogen gas. Next, the protein-coated stamp is placed with the structured surface in contact with the dried hydrogel surface, and brief pressure points are applied on the top of the PDMS stamp to ensure good contact between the stamp microfeatures and the hydrogel surface. The final step is to gently remove the PDMS stamp from the hydrogel surface and to clean the stamp. After passivation of the non-printed zones with VSA, cells can be seated on the micropattern hydrogel surface. Ultimately, an inverted fluorescence microscope is used to observe the protein microfeatures.
This method can help answer key questions in the mechanotransduction field, such as the relative contribution of extracellular matrix properties, for instance, stiffness, cell ligand density, or protein nature in the mechanotransduction process.
It is recommended that this procedure be performed in a chemical fume hood. Start by placing 25 mm diameter circular glass cover slips in a petri dish and smearing 0.1 molar sodium hydroxide.